Okay, good afternoon. Can you hear me, first of all? Yes? Okay. So it's wonderful to be here. It's actually my first time, I can't believe it. Last year I couldn't make it, but it's wonderful. I hear that the group is growing, which is important. I think for rare diseases, this is really important that you get together, and together you're stronger to make your case. You know, that there is enough resources to do science, and eventually pharma will help to develop therapies. So I'm, I have slides, I have a lot of slides, maybe too many, but it's also a bit technical, maybe too technical sometimes, but I will try to make it entertaining also. A bit like a genetic education, okay? So I'm a neurologist by training, but I work for the last 15 years really as a geneticist. And mainly I'm doing research. Okay, so let's do this. Well, first of all, I'm a geneticist, so you shouldn't be surprised. That's sort of my way of looking at things. Genetics really is everywhere. It's not everything. What you just heard the last hour is the other side of it. It's the environment. And the environment is pretty much, from a genetics point of view, everything else. But genetics plays a role in almost everything. Also, over the last decades, certainly with the rise of genetics as a science, you see these cover stories about all these wonderful things that are just around the corner, and they will change everything to the better. And there is a lot of overselling, certainly. But there is also a lot of enthusiasm. Usually overselling happens because people get so enthusiastic about something. So there's reason to be hopeful, and certainly I will hopefully show you some details where we made so much progress. And for instance, the Angelina Jolie story, which I'm sure most of you are aware, basically the fact that now we can do genetics on something like breast cancer, and that leads to really severe decisions for some women to make. That is a real impact on people's life. Not necessarily a very nice impact, but an important impact. So I think 5 years ago I probably wouldn't have showed you this slide, but increasingly I'm quite optimistic that something like genetic therapy will actually come, and sooner than most think. Just in the last year, there has been amazing progress on this front. So if that is true, then sort of it could be reduced a little bit to two sides here, like a coin, the HSP problem. One, of course, is what you're very familiar with is we have to make good genetic diagnoses, because the majority of the clinical problems many of you have comes from precise genetic defects. There are other factors, there's no question, but that's the root cause. And only when we can make these precise genetic diagnostics, we can then apply these upcoming genetic therapies, because they're going to be extremely specific. Okay? So, So I will talk most of the time about the one side of the coin, which is the progress we're making towards better genetic diagnostics. But in the end, I will show you a little bit where it's going with the genetic therapy. Okay, so I know very few of you are probably biologists, geneticists. Let me just give you an idea what the human body actually, why DNA is so important. First of all of us, our body, right, consists of cells that work together, a lot of cells. And there are about 40 trillion cells that make up a human. And most of these cells are very specialized: skin cells, liver cells, nerve cells, and so on. So a lot of cells that work together. And think about it, for all of us, it started with a single cell, right? We all started with a single cell, and then this cell divides into 2 and 4 and 8 and on and on. And we are the end product of this. So what is fascinating is that each cell, when it divides, it has information inside that tells how the daughter cell is basically an exact copy, and how that daughter cell knows how to make another copy. So it has that stuff in it that's called DNA, okay? And so the DNA is a pretty big sort of molecule, and every single of these 40 trillion cells, with some exceptions, has an exact full copy of how your body is being built, right? So it's sort of a hard drive that your computer has, or a chip that your iPhone or your telephone carries, that contains all the information. Every single cell has the complete set of information. And so this DNA, that complete DNA that tells a cell how to make a human body, is this long molecule, which, simply speaking, can be broken up into letters. We call them bases also. And there are 3 billion bases that make up the information that's needed to build a human body. 3 billion bases. It's a lot, but it's not endless, right? What's also interesting is all life on this planet so far carries the same genetic alphabet. Okay, there is no Chinese DNA versus American or German DNA. It's all the same language. That's, by the way, why biologists think that we all sort of evolved from lower animals into something that we are. So that's the DNA, and here's another schematic. You see a single cell here. The DNA is tucked away inside in this structure called the nucleus. And that's just sort of a schematic of how the DNA looks like. It's packaged in what's called chromosomes. And then when you go closer and closer and closer in a microscope, so to say, you see eventually these 4 different bases here of 4 different colors, okay? So you can imagine this is something so fundamental for biology, for medicine, for spastic paraplegia, that US government decided 20 years ago to decipher, to encode what the human DNA actually looks like, all 3 billion bases. And it took 15 years to do that. And when it was finished actually several times. The first time it finished in 2000. By then they had spent several $1 billion on this effort. And it was an international effort, not just a US effort. And you see here in the middle, of course, President Clinton and Francis Collins, who led this project, who's now the director of the NIH, Francis Collins. And on the other side, there is Craig Venter. So Craig Venter is sort of a private scientist. He has his own institute, and he managed to do the same work privately that this whole expensive government program was doing over 15 years. That's why he's in the picture, actually. So after this was done, in 2000, 2003, that time frame, a large number of studies happened that tried to understand how diseases correlate with DNA changes. Okay? So the first wave of these studies was something called the Genome-Wide association studies. Basically, what they tried to understand is how things like diabetes, heart disease, stroke, something that's sort of common, that a lot of people carry, Alzheimer's disease, how those diseases relate to DNA changes, okay? And they found, so this is a map, you see all these are the chromosomes we carry in our body. And each of these colored dots here shows one hit where they found a signal that there is a relationship with the disease, okay? So it was quite successful, these studies, and they're still ongoing. Now these studies include literally hundreds of thousands of people, right? To find statistically these signals. These are very expensive studies. A lot of patients have been included. So you would say, well, great, this is all being solved. But what we learned is, unfortunately, a little bit is, yes, you find a lot of these signals, and here's for diabetes type 1, which is just an example here. There are few signals, few genes, if you like, that carry a high risk, that are really bad for you. For instance, here this scale is the odds ratio. So literally what this means is if you carry this HLA, this first one, then your risk for diabetes type 1 is 7 times higher. 7 times higher. Now that sounds a lot, but maybe it's not so much. The chance to get diabetes to begin with is maybe, what, 1% or so? So now it's 7%. That's a lot more, but it's not like 50% or 80%. You still have a low risk, okay? So all the other genes you see here, they carry an even lower risk. Most of these carry a risk that is close to 1, okay? So the idea was always, okay, well, when you're really unlucky, you carry a lot of different risk genes, and then boom, you get diabetes type 1. But when you add up all these risks here, you still don't get a really high risk. So sometimes you read maybe in an article that with all these expensive studies, we can only explain 10, 15% of the risk that is there to get, say, diabetes type 1. So there's a big gap, right? We know there's more genetics there, but we can't quite find it with these studies. Now, in HSP, it's a little different. It's a rare disorder. The risk is a little different. If you have the gene, like spastic, and you have the bad mutation, you get the disease, right? And your child gets the disease. So the risk is super high. The odds ratio is super high. It's pretty much 100%. However, spastic, if you take 100 HSP patients, spastic explains 30, 40 of these patients. What's with the other 60? Oh, there are other genes like Paraplegia or REB1. These are other HSP genes. But those genes explain less families, less patients. And all the genes that we find these days, they tend to be quite rare and explain only a handful of families. So in some ways, this looks quite similar, and there is still a gap that we can't quite explain why some families, about 30% of patients, why we can't find a genetic cause, right? So that's the challenge we up again today, or right now. So this phenomenon we call sometimes the dark matter in genetics. It's just sort of a joke, but it means we don't quite know where this other risk is. Where are these other HSP genes, right? So that's what we're working on. So this graph shows you basically how expensive it is to sequence a human genome. And over time, and you probably have heard about this, about 18, 20 years ago, it was $100 million or more, $100 million to sequence a single human being, the genome, the 3 billion bases, $100 million. It has come down dramatically. It is now about $1,000 in just raw sequencing. So that improvement of technology really has changed how we do genetics. It has opened up huge opportunities to study the genes. And we will all see the benefits from it, I think. This graph is probably the single most important graph in genetics in the last 15 years. Things have become really cheap. They have become available, accessible. It used to be a very exclusive club that could do genome sequencing. Now you can do it everywhere in the world. Okay. So what do you see when you actually sequence a lot of people? So here's a study that came out a couple of years ago. They sequenced 60,000 people. It's like a humongous number. they used a variation of the genome we call the exome. Some of you have heard about this. It's sort of a shorter version of the genome. But still, 60,000 people, that's unheard of. And then they put all this data together. They were mostly people not with HSP, not with rare disorders, mostly so-called healthy people. And so what do you see? And trust me, genetics is actually simple. What we look for are simple things, right? So the first thing they look for, and that's the big takeaway here, is how many differences do you see between people? We also call them variations or mutations. Bad differences, we call mutations. They can cause disease. But a more general term for this is a variation. So how many variations do you see? They saw millions of variations in these 60,000 people, of course. But what was interesting is most of these variations, what is called singleton, means just they saw it just once. So most of the variations they saw only one time in these 60,000 people. And then the second most common type of variation, they saw only 2 to 10 times. In fact, 99% of all the differences between people are what we call rare. You see them only a few times. Okay? So, so what this means in some ways is means if you take just a random person like me, say, and sequence my genome and compare it to the data in this study, my genome will carry another few hundred variants that were never seen before in any other study. Okay? That's sort of how unique we all are. But that also means that for geneticists, it's quite difficult to decide which ones are the mutations, which ones are the really bad ones, because there's just so much variation around. There's so much sort of, in a way, background noise. Okay? Okay. So, in case you're interested, what is in a human genome? In a single human genome, which is again 3 billion bases, you find about 4 million bases that are different from what we call the reference genome. Okay? Each of us carries about 3 to 4 million differences. And in those of you who carry HSP, unfortunately, there's typically only a single one out of those 4 million that's in the wrong position, right? And that makes the disease. So of these 4 million, about 20,000 fall on genes in each single person. Okay? If you sequence a lot of individuals like these 60,000, they found 10 million variations. Maybe, and I already said, most of them, 99%, are really rare. What's also important is 85% of all the variation you find in a single person is very closely related to the genetic ancestry of this person. So you would immediately spot, without knowing the person, when you see the genome, the data, you would immediately spot this person is from China, this person is from Germany, this person is from, actually nobody comes from Pittsburgh, I guess. So, now why is this important? It's an interesting fact, and many of you may have done 23andMe or similar ancestry studies. They're sort of fun, but it actually has real medical impact. Because for the longest time, most research studies have included people that have an origin in Europe. They typically have a light skin, maybe blue eyes, so what we call Caucasian ancestry. But considering what I just said, people from other places, they have a different spectrum, a different set of variations. So this is a study that came out in a very prestigious journal, the New England Journal of Medicine, and is from a group of scientists and doctors who are probably the best in the world to diagnose a certain type of heart disease, an inherited heart disease, okay? And they have diagnosed people for for many years, 10, 20 years. What they did, they went back to their files, and based on these new data now, like the 60,000 people study, they went back to their file room and looked through every single report they had done in the last 15, 20 years. And they found a dozen or so reports where they had told these doctors and patients, this is your mutation. And in those dozen or so reports, they had to now say, you know what, based on the new data, that's probably not true. This mutation is not a mutation, it's just a variation. That does not explain the disease. Okay, that can happen, it's science, things have changed. What is actually concerning is all of these corrections happen in African Americans. Okay, because all the reference databases, they just looked at Caucasian genomes. Okay, so this is a country where you see people from all parts of the world. So that's a big challenge. We need to understand the genetics of the world to really understand every single disease. And that is also true for HSP. And it goes both ways, by the way. Understanding the HSP in China, say, or in Japan, or in Spain, actually informs how we understand the HSP in the United States. So another big problem in genetics is, okay, we can now sequence and test lots of HSP genes, and that's what we typically do, and you probably have gotten gene panels yourself. The more genes you test, actually the number of bases you test get bigger and bigger. Just by chance, every single one of us probably carries now a variant that could be a mutation in one of the many HSP genes that are typically tested now. So what this means is for your doctor is that when he does a gene panel, and it's a large panel, he almost always gets a report back that says, well, there is a variant here and a variant there, but we don't know what it means. We don't know if it's what we call benign, or if it's actually the disease causing. We call those variants of unknown significance. And unfortunately, they have become quite a plague, really, because it really hinders making good diagnosis. So that is a big topic. A lot of people work on how can we clarify which variants are really mutations and which are just there by chance. So. Okay, I'm jumping a little bit. With all the technology and all the new knowledge now, how well do we actually do? Now, about 70 out of 100 patients that come to your doctor, come to their doctor with HSP, about 70 out of 100 get a genetic diagnosis today. That's fantastic. And to put this in perspective, just think 20 years ago, when like Dr. Fink found Spastic first. Before that, there was nothing. And now it's 70 out of 100. But it's not 100, and it's not 95. So there is still work to do. And even with the most expensive sort of studies, these genome studies, or these short genome studies we call exomes, there is still a gap. We still can't find these remaining 30 patients, the genes that explain these patients. So then we're asking ourselves, what is this dark matter? Where do we find the remaining effect? And so a big question, of course, is now, should we really do whole genome sequencing? Okay, that's sort of the Cadillac of genetics, the whole genome sequencing. But it's also the most expensive study you can do. Typically, it's not being paid by health insurance companies. So, and that's something on the research front we deal with. Should we do a genome or not? In case some of you may have had this discussion with your doctor, it is true, you can actually find about 15% more information in a whole genome than in an exome. Okay, so it is worthwhile from that point of view. Also, the whole genome doesn't look just at genes, it looks at everything. And sometimes the mutations, they can actually lie outside of genes. So you have to do the whole genome then to actually find it. But even the whole genome is not what I'm trying to sell you here. There's always something that we are not telling you. Even the whole genome cannot sequence the entire genome, even though we call it whole genome. We shouldn't call it whole genome. There are areas in the genome, about 10–15%, that even the best technology today cannot really read, okay? And I'm happy to talk about this, but that's just also something. Even when you say, okay, I'm going to pay for a whole genome myself because I want to find out, there is still room where we can just not see today what is there. So, of course, scientists have tried to make the case for whole genome sequencing, basically doing more and more sequencing. This is one of these studies, not on HSP, but on severe intellectual disability. And when you do sort of classic gene panels or arrays in that disease, you can explain about 12%. If you do a whole exome sequencing, this is a more expensive test, in this particular disease, you can explain about a further 27% of patients. And when you do whole genome sequencing, you can explain even more patients, but not all of them, okay? So, another jump. So what do I work on? I work on HSP, as some of you know, but I also work on other rare disorders. So why doesn't he just work on HSP? It's so important. Well, the way I look at this is HSP is not a silo, okay? HSP is actually a very special case of a bigger picture. And what HSP really is, in some ways, is there are nerves in our brain that have very long extensions. We call them axons, axon. But there are other diseases where these axons are sick. But these are different axons, just not the types that get sick in HSP. And so there are these diseases that have problems with long axons from nerves. And we like to call them axonopathies, okay? They are diseases where these long axons get sick. So the inherited axonopathies are the ones that you can pass on to your children. And that's sort of the bigger picture I'm interested in. And there are other types, other names, like probably the prototype is the so-called hereditary motor and sensory neuropathies. And you may have heard this disease named Charcot-Marie-Tooth Disease. Very strange name, has nothing to do with teeth, okay? These patients, they have the neurons that are in your spinal cord and project into your arms and legs. Those nerves get sick. It's a very different, it's quite different, I should say, a quite different clinical picture. But from a biological point of view, there are definitely similarities. And altogether, HSP and CMT, there are over 120 known genes. Still, a lot of patients not diagnosed. And here's another view on this. So spastic paraplegia is here, CMT is right next to it, and you already see I sort of make this overlapping, because clearly, from many points of view, especially genetics, we know they are overlapping. There's another disease that's overlapping, ataxias. Some of you may have heard this. And so on. And so the point of this slide really is, again, no disease is an island, okay? It's all connected. And the genetics in the last 10 years has taught us how they are connected. And I think it's very important for you to understand that it's not just that you're not just each one of you is alone because you come together here to, be a group of people who have the same issues. But there's actually more than that. There are patients, there are groups just like you, like the CMT group. They meet somewhere else right now. But there is a connection. And some of the drugs that may come to the market in the future, I think they may actually benefit both groups, okay? So that's something to consider sometimes, how you can even team up with these groups in some fashion. Okay. So again, we are just part, my group is just part of a community of geneticists around the world who study HSP and CMT. And now we make fast progress, okay? It's a good time, actually, even though you don't get therapies yet. This is since 2012. These are all the genes here that you cannot read because they printed too small. These are all the genes that were identified just since 2012, okay? So there is rapid progress. That is certainly true. On the diagnostic side, we need to get better on the therapeutic side. And all the red colors here, I'm always proud to say, is where sort of we had our hand in my group. So that also means we didn't find them ourselves. We just have to be very collaborative. We like to work together with other groups. So one way to do that, to work together, we have actually, you know, considering I went to med school and now I do actually a lot of software development. So I changed a little bit. So we have created software, different types of software to help scientists to browse through genetic data faster, but also to look at genetic data together so that we can have genetic data in Miami and a collaborator, say from Germany, could look at the same data and confirm what our suspicion is. So that is what we do. We call this the Genesis platform. And this working together, we call sometimes also matchmaking, genetic matchmaking. So here are some of the more important groups that we have sort of convinced to come together in a database. It's the largest database for spastic paraplegia, exomes and genomes. This scales even higher. Databases like this have now teamed up and into something called the Matchmaker Exchange. That is to connect these databases in different parts of the world, and often they have very different diseases in them, to talk to each other and find overlaps. We have about 7,000 exomes and genomes right now from patients just like you. And the majority of these patients is currently not, we don't know the genetic cause. I mean, we're certainly biased to those that are not resolved. So we get sort of the hard cases, they come to us, the difficult cases. Here we compare in our data, variance in our database, all the variations we see to this big study I showed you, the 60,000 people. And I just want to point out one thing. When we compare these 60,000 people to the 7,000 people we have, about half of the variations we see in our data does not occur at all in this 60,000 people study. Okay, that's just how rich, in a way, how many variations are out there. To expedite this, actually, we have formed a foundation. We call this the Genesis Project Foundation, which helps connect these scientists all over the world and help maintain this infrastructure. And here's just one example how this works to make it a little bit more understandable. So my group, we have worked with collaborators for many years in Sao Paulo. And we have, one example is we have worked on two small families with a very complex clinical picture. and we came down to a gene, we thought this is the gene, it's a novel gene. But based on this one family or two families, we weren't quite sure if this is the right answer. There's another group in Germany where we learned they had worked on the same problem in also a single family, and they had come to the same answer. And because they both had their data in the Genesis database, we could actually match this data immediately. And so suddenly we knew there were patients in Brazil and patients in Germany who had the same answer. So we were quite certain this is a new gene that can cause HSP or that syndrome. And then in that data, in our database, we immediately found additional patients. Once we knew this is the one, we found more patients in France, in Germany, and also actually in Venezuela. So that particular syndrome we were so interested in is called Bucher-Neuhauser syndrome. There are like 10 patients were published ever, okay? So rare. And you can see it certainly doesn't look like HSP. It has other things in it. Ataxia, this is a brain disorder, an eye disorder, and also a problem with your hormones. Sorry. Oh, you don't see that. I get a warning here. So, but then we quickly saw that there's another syndrome, which people have called Gordon-Holm syndrome. And again, you see different things like ataxia, hormone issues, but then also spasticity. Now we're talking HSP, potentially, right? So, I tell you, scientists, doctors, we like to think about things for a long time. We give it weird names, and we're very proud about it, like Bucher-Neuhauser syndrome. Turns out the same gene can explain both super rare syndromes, okay? Frankly, I really don't care what they named. I just know this is the gene, okay? Because that is the unifying platform. It's the same gene. And when we then screened a large number of patients, we saw that it's probably too small, so I will tell you. We saw, so this is the gene. This is how a gene sort of looks schematically. We saw a lot of different mutations. So we saw patients with this Bucher-Neuhauser syndrome, Gordon-Holm syndrome. We found patients, they just had ataxia, nothing else. We saw patients that had just spastic paraplegia, nothing else. They would be like HSP. And in fact, Dr. Fink had published a few years earlier this particular gene as an HSP gene, okay? It's just that nobody else ever found another case, so we didn't know what to do with this. It turns out this particular gene, PNPLA6, can cause HSP, but for honestly unknown reasons, it can also cause very different things that sometimes can contain HSP, okay? So then we published it. A couple months later, this came out from another group. There is something called the Oliver-McFarlane syndrome, and another one's called the Lawrence-Moon syndrome. All fancy names, but they all explained by PNPLA6. So this is sort of how we try to show this. Here's spasticity, call it HSP. And then there are these other symptoms, ataxia, eye disorders, and so on. It's all the same gene, but for some reason, some patients, they have a number of these. They sort of span a spectrum. And sometimes it's just HSP, okay? It just also tells you again how these disorders that physicians have meticulously put into different drawers in different boxes actually overlap, okay? That's sort of the message. And of course, sure enough, a year later, another paper comes out, and actually it turns out PNPLA6 is a cause for a number of childhood onset eye diseases. No HSP involved whatsoever. It's so weird, and that's sort of some of the challenges we deal with. Okay, we have 15 minutes. I wonder, I'm probably gonna skip forward, show you the gene therapy piece, and then we have a few minutes left. Oh, what is the time? When is, until 4:15 or? Still 20 minutes left? 15 minutes left? Okay, I will keep going. It's fine.
You have time for questions?
Yeah, I want to definitely make time for questions. That's my point, exactly. So again, we talked about this already. We can find a lot of genes, but a lot of patients are undiagnosed still. If you consider this sort of the progress, we find we can explain more and more patients. Maybe we are here right now. There's still 30 to 50% of patients not explained. With all the new genes, maybe we get to 70, 80%. But it's possible that this gap that we really want to close is just there. And that's sort of one of the challenges. Maybe we are missing something. So that's another graphic to show you a little bit how genetics think about things. This is from a famous scientist at Baylor College, Jim Lapsky. He calls this clan genomics. Clan genomics. Basically, he says we're all part of a clan. Each of us, we have a clan. Most of us, we don't know our clan, actually. We used to, like 5,000 years ago. You used to know exactly what clan you're from. But the principle is the same. Say, this is sort of the now, and you see a family pedigree. You know your siblings, you know your parents. If you're unfortunate, you have HSP, and you know one of your parents has HSP, as an example. And then maybe some families know, oh yeah, grandpa also had issues, but then it sort of becomes quickly unclear, right? In most families. Some families go way back. So when you go back in time, you sort of go back into this pedigree, you go back into your clan, and it gets bigger and bigger typically. And over here where it's all sort of blue, that's all of us. That is your city, that is your, this is your state, this is your country. So it sort of gets lost a little bit in the overall population. Diseases like HSP that have a very strong genetic effect, we tend to see in pedigrees exactly what's going on, how it is being transmitted. But those diseases tend to, so these are the sort of strong effects. Other diseases like, say, diabetes type 2, where it's not just genetics, you also have to eat not so healthy, or maybe don't do enough exercise. It's not so clear how the transmission is, and so those diseases have weaker genetic effects, and you may not be entirely clear how to trace this back in time. And the point is, there are different ways to study these diseases. HSP we have studied with exome and genome studies, and we had great success. You wouldn't be so successful studying diabetes like this. The point is, I guess I'm trying to make is, to close the gap, this gap here, maybe we have to consider that there are weaker factors still, or that maybe two genes have to come together. And the place to look for those would be more in this middle field here, okay? So that's one of the things we're exploring, and we're using more statistical studies where we need a lot of patients to do statistical tests. One of them is called the gene-based rare variant analysis, and I don't really want to go into because of time, but that's a new type of study that requires a lot of HSP patients in one database. Another way we're trying to explore this gap is by modeling the genes we know already together with all the genes out there in gene networks. These are very computationally heavy studies. This is sort of a model of all the HSP genes. Now, there's really nothing to take away from this picture. That's why I'm showing you this. But if you then have a talented student, you can actually drill in and find some interesting pieces. So just from that network, what you find is, compared to a random set of genes in the overall gene network, turns out HSP and CMT actually statistically clearly overlap. So everything on this side here is related to each other. The genes have a relationship. Everything on this side, the genes are sort of random. They're not related. CMT, HSP, if you compare them, they clearly overlap genetically. So there is something here. Another disease where it clearly overlaps is ataxias. Okay? But not cancer, deafness, cardiomyopathies, which are heart diseases. Another disease where it somewhat overlaps is ALS, actually. And probably some of you have heard about this. Okay, so to summarize, I think what we need in the genetics field to keep making progress, it's becoming more and more about data. The sequencing itself is more affordable, is cheaper. We really think collaborating more and more with colleagues and acquiring more data will help us to maintain the pace of progress. And really the bottleneck is surprisingly not so much, one of the big bottlenecks is actually data analysis. And nobody wants to fund data analysis. We take it for granted. We focus on the vet lab, we focus on the sequencing, which is also sort of what we call vet lab. But we don't pay too much attention on the data analysis. It's kind of crazy. We live in a data world. Everything's data-driven, from Facebook to this. But we don't really acknowledge the role of data in medicine quite yet. And I think registries, and I know you're active there, are a good way to bring the community together and help the scientists. Okay, so just very quick to gene therapies. I think tomorrow you hear more about it. I do think personally that there is an opportunity here. We're not quite there, but something is happening there. So gene therapy, gene editing, which is changing genes, sort of the idea is, could we even correct the mutation in a single person at high fidelity? Okay? And you heard about CRISPR maybe? So CRISPR is simply a method to do that. I don't think CRISPR quite now will be the solution. There are certain risks with using CRISPR in a person. It could make, by chance, other changes, and some of those may actually cause cancer or other disorders. So CRISPR is not quite there, but it is heavily used and studied and constantly improved. There is now so much industry interest and industry, frankly, dollars, that I think CRISPR or whatever comes after will be here sooner than later. And again, this is sort of a small piece of the 3 billion letters, and then the idea is that you could actually go in and change single letters, like in a book. You would be an editor in a book. Now, there's one type of gene therapy that actually is already here. The first gene therapy was approved last year in the United States, ever. And it's for an eye disorder. It's not HSP, but it's for an eye disorder. And I have a little video here that illustrates how this works. And it would work quite similar in HSP, I think. So you would inject into the eye the drug, which is really a type of almost like a virus particle, which has the ability to find neurons, specifically nerve cells, go in there and then release a gene into the cell in each of the neurons. That's the gene that's missing or that's damaged because of a mutation. So it's a fairly straightforward looking process, right? And for 20 years, people have perfected it, sometimes with catastrophic effects in patients who had trials. People had died, actually. But it seems now in 2018 the technology is mature and can be used in patients. And I'll show you one example. So this is this company, Spark. I'm not affiliated to them whatsoever. But this is the first ever gene therapy drug. And this is an eye disorder. So this boy is, before treatment, he's blind. And he's asked to walk through this maze. And he can't do it quite. There is this big stop sign in front of his head. He doesn't see it, and in a second or so he will run right into it. It's just to show that he cannot see. He's legally blind. when doctors gave him this one injection in the eye, just one injection, 2 weeks later his vision was 20/20. He's going to normal school, no glasses needed. And this is stable for at least now more than 2 years. And here's the boy after treatment. He's just going through the maze just like that. So this is just, I call this almost a Jesus drug. You know, Jesus made blind see. It's unbelievable. And there's similar drugs now for spinal muscular atrophy, which now becomes quite close to what HSP is really. So, you know, it's happening. Something is happening there. And I think Corey will tell you more tomorrow about it. Okay, this is my team in Miami. Great team. And maybe we should use the time to answer some questions. Thank you.